Okay, you're welcome back. So let's continue. So here we have more terminologies. Now here we'll look at genetic cross diagram and the Punnett square. Now these are the two patterns we use in when you're trying to cross some traits. Okay, so the first one is what we call the genetic cross diagram. Second one is the Punnett square. Let's start with the first one. And most times in exam they will give you a question. They will be like, cross this, determine this using a Punnett square or using a genetic cross diagram and so on and so forth. So anyone they give you, you should be able to use any of them. Let me get my black. Oh, you work well. Okay. Now they've given us some examples here. And I'm going. I'm going to pick another example so that I can get the exact description I want. Now let me use the tall, the one that has to do with height. Let me use the trait for height. Okay, remember I told you height is a trait, while short or tall is a character. Short or tall is a character of that trait. Okay, so let's use this. So this represents heterozygous tall person, and here is a homozygous short person. Now think of this in the exam. They will be like examiner will be like, what will be the F1 generation of a man who is a heterozygous tall that got married to a woman who is homozygous short? So what will be the F1 generation? So determine the F1 generation of this couple. Now they will give you a question like this in a word format, and they will expect you to interpret it in a genetic format. Okay. Now they've mentioned that the man is homozygous, heterozygous tall, so this is it, and the woman is homozygous short, so this is it. So this one is homozygous recessive. Okay. Now using the first pattern, which is the genetic cross diagram, we'll go this way. So this will link with this to give you big T small t. Now this will link with this again to give you big T small t, right? Then we go again the second one. This will link with this to give you small t, while this will link with this to give you small t. So from this result, you can see that two of the children are homozygous recessive, that they are short, while the other two, they are heterozygous tall. Now there is no homozygous dominant here. There is no homozygous dominant here. So genetically, this is the gene, the genotype. While when it comes to phenotype, we say this person is tall. So this is the phenotype. This is tall. This is short. This is short. Remember, phenotype is the physical expression. So physically, these two children will be short, while these two children will be tall. Okay. Now, if I'm to let's look at the second one, which is the Punnett square. Now, if you are to use, if your examiner should request that you use Punnett square to do this, so you bring the parents. So you come here, you arrange the parents. You can decide to call this the male or female anyhow. So this is big T small t. I'll put it like this, and then I'll put small t here. So this is the man, this is the male, and this is the female. So we will draw our table. So this is our table. So this, we go with this to give you, I'll bring this down, big T, small t. Now someone will say, what if I write it this way? Now if you write it this way, it will still mean the same thing, but the best way is that the dominant always comes first. The dominant always comes first when it comes to writing it. So this will go with this, I will give it, this will go with this, will give you small t, small t. This will go again, this will give you big T, small t. This will go to give you small t, small t. So you can see now the F1 generation is same as this. So this is the F1, which we call the filial one. Okay, filial stands for son, children. Okay. All right. So I'll give you a task now. So you're going to pause the video and do this task. So this is the task. So the first one, you're going to cross these two traits I'm going to give you. So let me use traits for eye color, big B, small b. That's the blue, brown being dominant, and the small b being the blue, which is recessive. I have this and this. Then number 2, let me use this. This is for white, and this is for black. So this will cross with this. So cross this for me. Pause the video. Cross this, and if you don't understand it, play the video back and understand it. After that, cross it, and then drop a comment for me on the comment section if you've gotten it. But if you've not, if you don't understand it, even after playing back the video, you can also ask me questions from the comment using the comment box. Sorry. All right. So like I said, pause the video and do this task. So this is the first. This is task 1. So pause the video and do the task 1, and then after that you can play on the video. All right, so having gotten this, so let's move on to the next terminology. The next terminology is hybridization. Now from the word hybrid. So what are hybrids? Now hybrids, they are the F1 generation that is being produced as a result of two contrasting characters, two parents with a contrasting characters. That's what produce hybrids. Now hybrids, think of animal hybrid. When you bring a Rottweiler and cross it with a German Shepherd, it's going to give you something that looks like both, but it's neither German Shepherd or a Rottweiler. Think of the same thing. Your parents, your dad has his own character and traits. Let's assume that your dad is tall and you married your mom. Your mom is not that tall, or your mom is not tall. Now you can see that both of them, they came with two contrasting characters. Both of them will now cross to form you. So when they form you, you look like your dad and your mom at the same time. Now you're not going to look like your dad alone. You're going to have a feature from your mom and your dad at the same time. So that's why you're a hybrid. So we are hybrids. Now hybrid does not necessarily mean who is a cross between, let's say, a human and an animal. No, that's not all that is like that is about hybrid. Now the crossing can be from similar species, and the crossing can also be from two different species. Okay, so any of the two. So hybrid is hybrid. Hybrid simply means a combination of two contrasting characters that form one. Okay, I hope you understand that. Now this hybrid can be monohybrid, it can be dihybrid. Now when you talk about monohybrid, from the word mono means one. Now when you talk about one trait, let's look at this man. Now this man is let's look at his height and his skin color. Let's use skin color. Now this man's height is what we refer to as a monohybrid. Now why is it called monohybrid? Now monohybrid means that you are referring to one trait. We're referring to one trait. Now the trait here is height. So I'll call this is the trait. Now the trait here is height. Now look at another trait here. The skin color is another trait. So this is also another monohybrid. So this is another trait, which is skin color. Now these two traits, when they come together, we call that dihybrid. Okay. Now look at some example. Now let's say this man is for height. This man is tall. Let's say this is the type of tall this man is, and the man is brown skin. Let's say this and this. So this is a typical example of a dihybrid. Now I'm going to circle this two together, and I'm going to circle these two together. Now reason is because each of them represent one trait. Remember, I told that alleles occur in pairs. Okay, so this is the first allele, this is another allele. So these are the two traits. One is for height, the other one is a single. So we say this person is a tall black man, or this person is a tall white man, or you can say this person is a short black man. You can say this person is a tall short black white man. Okay, so that's what we meant by monohybrid and dihybrid. Monohybrid is when you deal with one trait, or when one pair of traits are crossed. But the hybrid is where you talk about two pairs of contrasting characters. Now here, looking at this example, we have yellow round seed. This yellow here is what it represents with big letter Y. Why this round seed is what it represents with big letter R. So that's why I have this is yellow and this is round. Same thing for rain code. So small letter Y. Remember I told you you don't use, let's assume you are dealing with height now. So this person is tall. Because this person is a carrier of short gene, this person is a heterozygous tall person. Some will say let's put S here. S stands for short, while T stands for tall. No, it doesn't work like that. Anytime you're using a letter, use a smaller version of it to represent the opposite. Okay. If you want to use S, you can decide to use S as well and use small letter S to represent the opposite. So you don't change the letters. If you're using B for brown eye, you're using small letter B for blue. If you're using big capital letter W for white, you're using small letter W for black. Okay, so that's the rule. So don't is not necessarily that it's going to be the first letter of the word. No, you can use any word. That's why there's always a room for keys. Always show your key. Key means I tell them, okay, this B represent this one represent that. Okay, in the exam, if you're giving that in the exam, you will also be given a key for you to know what each of them represents. Okay, all right, so I guess you understand that. So let's move on to the next terminology. Now next terminology is F1 and F2. Now I've told you before, F stands for filial, F stands for filial, which stands for son. Now F1 is like saying first generation. So these two parents cross to get this first generation. So this is the F1 generation. So this one produces recessive parent and so on and so forth. When you bring down one parent and cross it with the F1, it will produce the second, the F2. So that's how it works. Now think of an animal. Think of a goat. A baby goat, I don't know what they call those baby goats. Now baby goat is going to grow up to become an adult and then start mating with the mother to produce other baby goats. Okay. It goes for animals. It don't necessarily work for human beings, but it goes for animals. So when you say using crossing the F1 with the parents, that's what it means. Okay. All right, look at this terminology here called the incomplete and codominant. When we talk about incomplete, look at the English word. Let's start with look at the two images you have there. Look at the second one. Let's look at the second one. The English word co-dominance. We've already achieved what dominant is. We said alleles can either be dominant alleles or recessive alleles. And we said dominant alleles always overshadow the recessive one, and dominant alleles are always written in capital letter. So anytime you see any letter in capital, then cap, that means you're referring to dominant one. Okay. Now, if you look at these two images here, you can see one is talking about co-dominant, meaning that the two of them are dominating. Now there are different co-dominant alleles we have in nature. One is the blood group. Those whose blood group are AB, these are co-dominant. Okay, same thing with XY and XX, these are co-dominant as well. Same with the SS. Which other one? So think of different alleles that we have in nature that are co-dominating. Okay, so this meaning that this picture depicts a co-dominant allele expression. You see, this is when both of the alleles are expressed in a heterozygous. Now this is a pure red. You can see the alleles are dominating because they are both written in capital letter. This is the pure white again. Both of them are both written in capital letter, so they are dominating. Now they have their F1. This is the F1. Now F1 picked one from the male, one from the female. Now you can see the spotted cow because he picked that from the parents. Okay, so that was all called co-dominant when the two alleles are expressed at the same time. So that's what we call co-dominant, when the two alleles are expressed at the same time. But when it comes to incomplete, now this is when the traits are neither dominant or recessive, and the offspring exhibit traits that are a blend or mix of two parents. Now you can see this is the parent A. I'll use word parent 1, parent 2. Now parent 1 is a homozygous dominant. It has the homozygous dominant allele, while parent 2 has a recessive homozygous recessive alleles. Now, when they give birth to their child, you can see this A will cross with this smaller a here to give you big A and small a. So this child, the offspring here is a recessive, is a heterozygous, is heterozygous because the two alleles are not the same. Now, if you look at the two, you can see that there's no key here, so we don't know what this A represents. So we assume that this is for color. We assume this A to stand for color. There's no key here to tell us, okay, this is what this represents. So we assume that this is for color. So by the time red mixes with white, it will give you this color. So this, in this case, we say the dominance here is incomplete because the offspring looks entirely different from the parents. Okay, now when you take this F1 again, this is F1. When you cross this F1 with one of the parents, this P here stands for parent. When you cross F1 with one of the parents, now probably looking at the F2, you can see that what they cross is parent 1. Parent 1 was what crossed with the F1. So when you cross F1 with parent 1, it will give you the result. Somebody will say, why? How do you know? How do you come up? How do you know that is the first one? Now look at the result. Look at the first F2. There's no way if they had used the parent 2 to cross with F1, there's no way the other A is coming. There's no way the other A will come into play. So that's why we have parent 1. Okay, so look at the children. So they produce children that are the dominating alleles is incomplete. So that's what we call incomplete dominant, when the traits are neither dominant or nor recessive. Okay, so you can see now the result is something entirely different. There is not dominant, it's not recessive, something entirely different. Okay, all right, so moving on, more of terminologies. Now next one, we have the backcross. Now this backcross is the cross of an F1 hybrid to one of the homozygous parents. So when you say F1 hybrid, I believe you understand what hybrid is now. Now look, coming back to this image, you can see this F1, this is a hybrid, this is also another hybrid because they are carrying both their parents alleles. Okay, so I said hybrid can either be a crossing of two contrasting alleles or two contrasting characters from parent characters. Okay, it can be of same species, it can also be of different species. So you can have a hybrid of same species, you can also have a hybrid of different species. Okay, so when you say backcrossing is when the F1 hybrid is being crossed with one of the homozygous parents. Now if this is the case, when I say homozygous, from these two pictures we have here, both parents are homozygous. This one is homozygous dominant, this one is homozygous recessive. This one is also homozygous, this one is also homozygous. When I say homozygous, referring to the fact that they have same alleles. The two alleles are the same. That's what we call homozygous. The word homo means similar. Okay, so the two alleles are the same. So backcross is when the F1 is being crossed with one of the homozygous parents. So it can be parent 1 or it can be parent 2. So that's backcross. Now when it comes to test cross is more specific. Test cross is the cross between uses the homozygous recessive parents. You are using the homozygous recessive parent. So if that's the case, you can see now parent 2 is what qualifies. Parent 2 is one with there is a recessive parent. There is no homozygous recessive in this order in codominance because the two alleles are dominating. There's no recessive allele there. So, but in parent 2, in the other picture, parent 2 is recessive. So test cross is where you use a homozygous recessive parent, while back cross is where you use any homozygous parent. Okay. Then next one is mutation. When we talk about mutation is the altering of the gene as a result of environmental factors. If you look at a natural selection, you see that environment can bring about an alteration in the gene of organisms. That this happens over time though. So when they stay too long in a particular environment, they're going to develop a survival mechanism that affects their gene and changes their gene. Okay, and once that happens, those genes now can be transferred to their offspring as they grow. Think of if you go to the Antarctica and you see a polar bear, all polar bears that live within that region look the same. So they develop all those characteristics as a result of the environment. Now they use that to survive, but after surviving, they ended up keeping it and transferring it to the next offspring. That's why all bears you see, polar bears you see within the polar region are all They have specific characteristics. There is large, large force, and then they also have um, fats that helps them keep warm. Excuse me. Now, next one, selective breeding. Selective breeding is where you intentionally pick a particular species, and crosses it with another one. Let's assume you want to get animal white skin bull. So you're not going to bring the black. If you bring black, there's going to be a co-dominance here. So what you do is that you bring another white. So that's what called selective. You select them based on what you want to produce. What you want to produce as offspring. So it's just called that selective breeding. You pick them up and you select them, and then you breed them together. The last one here is the sex linkage. This sex linkage is also known as X-linked. Now, sex link simply means when something is being carried on the X chromosome. Think of in male we are XY, female are XX. Now let's assume, let me pick albinism for example. Now this is male. Let me put this albinism here because albinism is X-linked, so it's always on the X chromosome. Now when it comes to female, now this albinism, some of these diseases can be recessive disease or homozygous recessive disease. Some can be homozygous dominant disease. Now what it means is this: you remember I told you this person is tall, despite the fact he's a carrier of short gene, but this person is short. The only time this feature will reflect is only if the two alleles are recessive. So in the male, if they have one, this particular male here is albino, will be an albino. Okay, but this one stands a chance of not being an albino. Why? Because there's another allele that can contrast this particular allele. There's no another X allele that can contrast this, or X chromosome that can carry another allele that will contrast this. But in this case, now let's assume another one come into play here, a dominant one. I don't know what this is. This can be anything. So once this thing come into play, it will block this albinism from showing. But in male, you don't have any other X that stand a chance of carrying a dominant allele that will block this one. So in the case where you don't have a dominant allele that will block this one will always manifest. You see it now? So that's what we call sex link. When the disease or anything or any of these things which are carried by X chromosome is being carried by X chromosome, let me put it that way. So example, we have color blindness, we have hemophilia, we have baldness, we have sickle cell anemia, we have albinism. Now, some people make the mistake of saying that this is sex-based. No, in fact, male stands more. There's more. This is how I put it now. Like from what I just described now, there's more chances of this sex link not to show in female than in male. So if one recessive gets into the male it will show. Okay, but when it gets to the female, there's chances of it being overshadowed by a dominant one. Okay, so but there's no chance of such in male. Okay, so that's for sex linked. As I said, when the X chromosome, the character is being carried by the X chromosome. Okay, that's what we call sex linked. Okay, so let's talk about this guy called Gregor Mendel, his work. Now, let me give you a full overview here. Then okay, let's leave it for the next class. I don't want the video to be so long, so I want to have it in a short format so that you can easily access it. Alright, so next video we'll talk about this Gregor Mendel and his work. Once again, stay tuned, don't go anywhere, because I'll be right back.